Hello, and welcome to the Solid Phase Peptide Synthesis series made by Dr. Naomi Lee's research group at Northern Arizona University. In this video, we will be demonstrating how to fill out the peptide synthesis sheet. First, you will locate the most updated Excel sheet from the lab OneDrive. Download your own copy and open it in Excel. You will need to fill out all the areas highlighted in yellow, starting with your sample name, sequence, and start date. You will need to calculate the molecular weight of your peptide using either the ChemDraw program or GeneScript.com, as demonstrated here. In GeneScript, you will type your sequence out and add any modifications needed. In this peptide, for example, we are adding lipoic acid to the N-terminus. Once you submit the sequence, you can copy the molecular weight and paste it in your spreadsheet. You should also update the resin type or scale on the third row if needed. Now you will calculate the amount of amino acid used during each coupling. Serine is inputted already as an example. You can see that the formula is equal to the equivalence times the scale times 0.001 times the molecular weight of the amino acid, which is listed at the bottom of the sheet. Repeat this formula for the rest of the amino acids. Now you can fill in the sequence boxes. Start with the first amino acid you will load onto the resin, which is the C-terminus of the peptide sequence. Then, work your way from the C to N terminus down the sequence. When you are finished, it should read backwards from what you wrote in the sequence box. To print the completed sheet, remove all yellow highlights. Next, you will highlight from cell A1 to U44. Under Page Layout in the toolbar, select Print Area and click Set Print Area, then select Narrow Margins. Before printing, double-check the preview and make sure that everything fits on one page. Once you've printed this sheet, you should attach it to your notebook. This will be your checklist and cheat sheet for hand synthesis. As you do each step, check off the box so you can remember where you are in the experiment. The shaded boxes are for the anhydrine test, which is used to confirm that our coupling and deprotections are working. Whenever you reach this step, record your color results as P for purple or C for clear. During the steps that include weighing out the resin or the amino acid coupling components, refer to the mass table on the right. The solutions used in peptide synthesis are listed at the bottom of the sheet, as well as how to make them and what they are used for.